Hope everyone able to see my screen. Okay. So yesterday we started discussing about uh,
Recording in progress.
about the drug, about what are the clinical trials we have, and how these medical companies will reach out to FDA and how the CDs was evolved. Correct? And we started discussing the background process for these clinical trials. Okay. So in the process, the first thing we discussed about the protocol document. Okay. Sponsor will provide the protocol document. This we saw it yesterday an example, sample protocol. The protocol will contains everything, all this, how many subjects was involved. Okay, what is the primary objective, secondary objectives. Okay, the subjects will be screened, how it will be screened, some eligibility criteria, that is inclusion criteria, exclusion criteria. Okay, in each visit, what are the tests we have to do it. All this information, all the, about the clinical trial information, it is available in the in the protocol document, which will be provided by the sponsor. Okay. So in this process, the next important document will be CRF. Okay. CRF is nothing but case report form. Case report. Okay. So what is this case report form? So generally, if you go to any big hospital, when you go immediately, they'll give one form to fill you. Right? What it contains? Anyone observed? What the form will contains? The form will contain your name, your date of birth, age, sex, previously have any medical problems, any allergies, any surgeries, any information. All the details they ask you to fill you. Did you observe? Same way in the clinical trial process also, for each subject, we are going to maintain one form, that is case report form. Let me show you a sample. So this is a case report form. So for every subject, we are going to maintain one form. Okay, so this is only very, very important thing. Okay, because every information will capture in the CRF form. From the CRF only, it will come to data sets. Data sets only will prepare the, will write up programs and will generate the reports. Okay, so this is at hospital only, they'll prepare, they'll collect these forms. So this form only is the data here, everything. Okay, now let us see in this form what are the information we are collecting. What is the meaning of each one? Okay, the first thing, whether first subject will be screened, whether it will be eligible or not to participate in the clinical trial. Every study have list of inclusion questions and exclusion questions. He has to answer. Is a subject healthy male aged between 30 to 80 years? Yes or no? If we click yes, he is included. If we click no, he won't include. He won't eligible. Similar for all exclusion. If a female be a pregnant, if she click yes means we have to exclude her. If we click no only, if we tick no only, yes, we have to add her in the study. Like this, subject has to answer all these inclusion and exclusion questions. Based on that, we'll capture SR. Okay, first. So like the first page, inclusion and exclusion criteria. Next. when you come for the first visit, visit 1, at the time of screening. Okay, so first at the time of screening, has the subject freely given the written the informed consent? Yes or no? What is informed consent?
Giving willingness to
Can you clearly explain? Some voice was breaking. What is informed consent?
giving willingness to participate in the study.
Correct. So simple. Informed consent is a legal document. It has to signed by the subject. In the legal document, it will be mentioned, okay, saying that, okay, these points we are responsible, these points we are not responsible. If you are agree with this one, please sign. If he signed only, we'll take him into the consideration. We'll start our clinical trial process. That is the first step we'll do it. Okay. So that one. Next, once he signed only, we'll capture the screening information. So we'll ask the subject, what is your demographic information? Like what is your age, sex, what's your height, weight, body mass index. Okay. See here. Do you have any habits? Like smoking habits, alcoholic habits. So this information also will capture. Next. So this is a separate page. Previously medical history. Subject have previously any medical problems, have any disease or any illness or any surgeries, are you met with any accidents? Previously have any medical problems. That medical problems will capture in this page. Previous medical history. In the past, if you have any diseases or any medical problems, will capture here. Okay. Next. Physical examination. So we'll start doing the test now. Okay. All type of tests will do it. First, physical examination. Physical examination is nothing but all the body systems will check. Like how is his heart functioning, normal or abnormal? His kidneys functioning, normal or abnormal? How is his skin? Skin have any, it's normal, have any rashes or something? It's normal, abnormal. If abnormal, what is that abnormality? Will capture. Like this, all the body systems will examine the normalities and abnormalities will capture. Next. Vital signs, pulse rate, blood pressure, systolic blood pressure, diastolic blood pressure will capture. Next, we'll do the ECG test. We'll do the test and test results will capture. Next, lab. The laboratory. In the laboratory, there are 3 major category lab tests will do it. Hematology, clinical chemistry, that is urine analysis. These 3 categories, under each category will be having different tests also. So all the tests will do it, and the test results also will capture in this page screen. So all these tests will do it at the time of screening. When you come for first time visit, right? All this information will collect, demographic information, and all this lab test will do it. Those test results will capture. So now the same test will repeat when you come for a second visit. We told him to come in the next visit, is in next 1 week. The week 1, you have second visit. So when again he'll come for a second visit, yes, all the tests will do it again. All the physical examination tests, vital signs, laboratory tests will do it, and test will do it, test results will capture. Same will repeat again. When you come for third visit, his third visit in a, let us assume in a week 26. Again, when you come for third visit, we'll do all the lab tests. Same repeating again. Physical examination, vital signs, laboratory details. If required, EEG also, ECG also will do it. Week 4. Physical examination, vital signs, laboratory information. Visit 5. Repeat. So remember, every visit will do the tests. Why? If you do the test only, right, in the parallelly will take the drug. Okay, whether the drug is working or not, how can we know? By doing the lab test and by seeing the lab test results, we'll get to know whether the drug properly working or not. Correct? So parallelly, we'll take the drug and we'll do the lab tests. The test results will capture in the CRF. Okay, in every visit it will be the same. Apart from C, you have separate pages. Concomitant medication. See, this is important. Concomitant medication is nothing but if subject is taking any additional treatment, any additional medication apart from our study treatment. So in this clinical trial process, subject has to take, suppose for us, your study is about some drug A. He is supposed to take drug A only. Apart from drug A, if he is taking any other tablets, other medicines, other medication, if he is taking, what medication is taking, we have to capture here. Okay. Generally, we will take the medication in two scenarios. Scenario one. Previously, subject have any problems. For example, previously subject has some asthma or BP or sugar. We have. For that one is taking the tablets. And the same tablets is continuing after our clinical trials are started. Now that tablets information we have to capture here. For example, he's taking some BP tablet. How much dose is taking? How many units? Means how many tablets? 2 tablets or 3 tablets is taking? What is the reason when he's starting, when he stopped, is still continuing or not? Okay. So this information will capture. This is scenario one. Previously have any medical problems, due to that he's taking an extra medication. The details we have to capture. That's a serious scenario one. Scenario two. Clinical trials have started. After the clinical trial started, he got some side effects. Let us assume we have got some fever. For a fever, it took some Dolo tablet. That information will capture here. Due to side effects, that is due to adverse events, if he's taking any medications, extra medication, that information you have to take, you have to capture it. So in these two scenarios, we'll capture the details in a concomitant medication system, in the concomitant medication page. Okay. Next. Your separate page, adverse events. Adverse events is nothing but side effects. Once the clinical trials are started, this is important. Okay. What is the side effect you got it? When it is started, when it was ended, how severe it is, what is the final result? You recovered or not recovered? All this type of information will capture in a adverse event. There is a separate page you have. Finally, end of study form. So here we'll capture the status of the subject. The subject completed the study or not completed the study, when discontinued or any death was happened, we will take here the status. Clear? So this is the information you will be having in a CRF form. Okay. Previously, these CRFs are used to maintain in a paper CRFs. Okay. I told you that this clinical trial process, it will take some 2 to 8 years of time. So maintaining of 2 to 8 years of time of these papers, maintaining is so difficult. Now everything was digitalized. Now they are maintaining eCRF, electronic CRFs. Okay. Same form it will be available in a online. Okay, clear? So this is the CRF, second important form. Clear now, understand what information you have in a CRF? Now what immediately we'll do? So now the background teams will come into the picture. The first team involved will be CDMT, Clinical Data Management Team. Clinical Data Management Team. So in this team, there are two category people will be there. One is data entry people. Data entry people and data managers. Data entry and data managers. Okay. What these data entry people will do? These data entry people, okay, they're going to refer the CRF forms and the paper CRF or eCRF. They'll refer the CRFs. What they'll do? They will prepare the raw data sets. Means all the CRF information they'll capture in the form of data sets. Whatever in the site for each subject, they're maintaining the CRFs, right? All the CRFs they'll collect from the CRF information. They'll prepare the, for example, you're working for one project. So they'll prepare the raw data sets like this. See, that is all the subjects, adverse events information, they'll capture in one raw data set. Like this, see, all the subjects, medical history details, one raw data set. All the subjects, some information, see, informed consent information. Like this, they will capture in the form of raw data sets.
So they write in SAS data set, sir.
Yeah, they'll give in the data sets only. Directly they'll prepare the SAS data sets only.
Directly they prepare SAS data set.
Yes, they'll prepare the SAS data sets.
Okay, sir. Could you please open that one, sir? One.
Yeah, I'll open anyway later. In the project, we'll see that one.
Okay, sir. Okay.
So now this raw data sets only. In the next team is our SAS team only. Is input for us.
Yeah. Okay, sir.
So we have to take it. These data sets are the input. We have to use this no format error to open these data sets. Okay. See. A is one raw data set. They are prepared for. So all the subjects. See, all the subject. What are the adverse event name? It will be having like this. Okay.
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 Okay, sir.
So a particular adverse event, when it started, when it is ended, the dates, every information in the specification or in the CRF, whatever the information you have, they have to capture in the form of data sets. Okay. So that is the main data entry persons will do. Okay. Next, data managers, what they'll do. So, okay, data managers, they will validate the data entered by the data entry persons. They will validate the data entered by the data entry persons. Okay, because it's very, very important. Okay, this step. Why? Because if the CDM team, if they entered any wrong data mistakenly, anything, if you enter anything raw data, anything while preparing the raw data set, so down the team, all the teams will affect. Because from this raw data sets only, down all the team will started working. So your final reports also will affect. So listen carefully, these managers will validate and they'll send this raw data to next our team only. Okay. So next after CDMT, the next team is our team, SAS team. So remember our SAS team and the biostatisticians will work together. Okay, the statisticians will work on SAP. We both will work together. Together means, so these biostatisticians will tell our SAS team saying that what are the reports you need to prepare. What are the reports they are required. So those reports only will prepare and will give to them. What are those reports? So these only SGTM, Adam tables. Okay, so what biostatisticians will do? They'll work on SAP, right? SAP is nothing but statistical analysis plan. Okay, what these people do, okay, they will do the analysis on the protocol. Okay, they'll go through, they'll understand the protocol for each study. For that study, what are the reports was required? Okay, they'll inform us SAS team. Okay, so these biostatisticians, these people will do the analysis. How this analysis plan they will prepare? This one prepared based on the protocol document. Okay, so what was the end of the meaning? Your biostatisticians will clearly tell. Biostatisticians inform the SAS team what reports they are required. So they'll inform to SAS team. SAS team will prepare those reports. What are those reports? This is only SDTMs, Adams, and tables. Especially their work is mainly this one only. SDTM, Adams, anyway, they have standard CDs, the rules will be there. Mainly for TLF, tables and listings, what tables they require, what listings are required. These biostatisticians will give the mock sheets, okay, what tables, what listings. Anyway, we'll discuss separate chapter. TLF at last. So biostatisticians will tell, okay, these tables were required, these listings were required. Now those tables, those listings only will prepare our SAS team along with SDTM Adams. Okay, so now SAS team, by taking these raw data sets which are provided by the CDM team, will prepare all these reports.
That means we are not using the protocol, sir.
Protocol will use for reference. Obviously, we have to go through the protocol.
Okay, that means a newspaper reading of protocol is important. That's all.
Just a one-time reading is enough.
Okay.
The protocol information also will capture in the SDTM. There is some domain trial design models will be there. So
Okay.
they will capture. Okay. So that's also important. More important is the CRF form. Okay. We have to make sure every information was captured in the CRF. We have to capture in the datasets. Okay, especially in SDTM datasets. Going forward, we'll understand this one. But as of now, just understand the flow. Here, the flow is important. Whether the teams was involved is important. Okay. Going forward in detail, we'll discuss later. That is different case.
Okay, sir. Okay.
So first CDM team, they prepare from the CRF, they'll prepare the raw data sets. Again, next is SAS team. They'll work with the biostatisticians. We'll prepare these reports. The next team is medical writing team. Medical writing team. Okay. So what this team will do? So this medical writing team, they will, by referring these reports only, we are submitting the reports. So they will write a synopsis. Synopsis for the reports. Synopsis is nothing but So the overall, can I say the overall summary of all the reports? Overall summary. Are any derivations? Are any major side effects, etc. Synopsis is nothing but the overview, the understanding from the reports. This medical writing team, they'll write and they'll give. Okay. Once the medical writing team, they are also completed their synopsis. The final team will be statisticians. These statisticians, they will prepare the final document. CSR, clinical study report. Clinical study report. This final CSR document only, they will send to FDA. But remember, to the FDA, not only CSR document, along with CSR document, all the teams data, okay, from the CRFs, okay, CRFs we have to submit. CDM people, they are preparing the raw data sets, those things we have to send. Okay, now the SAS team is preparing the SAS reports. And medical writing team, they're writing some synopsis, that one. And finally, along with it, CSR. All things will send to FDA. That's it. With this, work going to be complete. Clear? Understand the flow from where it is started, from where we reached FDA. So first the thing, start with the sponsor. Sponsor will give the protocol. Run the protocol. Subjects will be recruited and investigator will be recruited and the process will be started. Okay. Then for every subject, we'll maintain the CRF information, CDM team. Next is CDM team. CDM team prepares the raw data sets by referring the CRFs. Now by next is SAS team. SAS team and biostatisticians will work together by taking the raw data sets as input. We'll prepare the SAS reports. Next, these reports will be referred by your medical writing team. They'll write some synopsis. Then finally, statistician will review all the reports and synopsis. He will write a CSR document. That CSR document they'll send to the public. Clear? Any questions, anyone?
So PPL means
What is it?
PPL.
PPL.
Peoples.
People.
You have write that PPL, sir. These people do analysis, na.
Okay, people. Yes. Okay.
That's
Statisticians people will do the analysis.
Sir, these people, that means the medical writers, what they will do, whatever the report we have given, they will write, sir.
That's it. In a whatever is SAS team, they are preparing the reports, right? By referring those final tables and listing, especially, they'll refer that one only. By referring that one, they'll do the writer synopsis.
Okay. In entire work, we are the main people.
We are the main only. We are SAS team only in the total process. We are only the main thing. Whatever we are submitting the reports is important. By whatever they are submitting the report, that is only from based on the reports only, medical writing team will write and statistician as well write a report. They'll write summarizing, okay? They just, but they are referring our document only. So that's the reason whatever we are submitting is important. Okay. Fine then. So I got an idea. Now let's discuss. The next topic is. I told you yesterday I'm going to give some definitions. So we'll discuss some definitions which we require to know to read any protocol document in the further process. Let's discuss about some definitions. First is study design. There are two types of study designs we have. Parallel study design, crossover study. Crossover study design. So your study for protocol, if you go and read the protocol document also, it will be mentioned whether it's a parallel study design or a crossover study design. So what is the meaning? If it is mentioned a parallel study design means, when we call the parallel study design, in a clinical trial, if a subject will be exposed to only one treatment in a clinical trial, the subject will be exposed to only one treatment, and the same treatment they will be continued, and the same treatment will be continued until the treatment end phase. Let us assume in your study there are two drugs are there, drug A and placebo. So for example, 101 subject is there. 101 subject, if you take the drug A, the same drug A, he will continue until the end of the treatment. You take 102 subject is there. If he's taking the placebo, the same placebo, he will continue until end of the treatment. If it is taking like this, it's a parallel study design. In a clinical trial, subject will be exposed to only one treatment and same treatment will continue until end of the treatment. Okay. Clear? So this is parallel. See, parallelly, one person will take drug A, one person will take a placebo. What is placebo? Anyone idea? What is placebo?
Plasma is nothing, is
one kind of drug.
a kind of drug which do not having the API, sir. That is the plasma.
That active ingredient will not be available in the plasma.
That's the
It is a sugar-free drug. Okay. The more major active ingredient will be there. It's a normal tablet. Okay. And the drug A is the letrozole drug A is the, it has an active ingredient which you are testing. Okay. Next. So how this information in a data set level will capture? Subject ID, arm is nothing but the treatment. For example, 101 subject is taking the drug A. Like this will capture. 102 subject is taking the placebo. So like this will capture in a data set level for a parallel study design. Now come to crossover study design. Okay, here the subject will be exposed to multiple treatments. Here the subject will be exposed to multiple treatments. For example, subject is taking drug A. let us assume in your study, there are 3 drugs are there. Drug A, drug B, and placebo. Let us assume 101 subject is taking drug A. After some days, is taking drug B. After some days, is taking the placebo. Similarly, 102 subject is there. Let us assume first is taking the placebo. After some days, is taking drug A. After some days, is taking the drug B. So observe here one subject is taking multiple treatments, drug A, drug B, placebo. 102 subject is taking placebo, drug A, drug B. If we will take like this, it's a crossover study design. So I was telling like after taking the drug A, after some time we'll take a drug B. After some days, some weeks. So that some days, sometime we call as a washout period. Some people will also call rest period. The meaning of the so clear. Washout is nothing but after drug A, after he's taking the drug A has to completely wash out from his body, then only has to take the drug A. Then you'll take the drug B, correct? Or else, after taking the drug A, take some rest, some time, give some gap, then take the drug. Clear? So the time between the drug A and drug B we call washout period or a rest period. Okay, same thing. After the drug B, have some washout period time, then it will take the placebo. Same will happen here as well. For 102 placebo, some washout period, then drug A. Drug A, washout period, and drug B. Okay. How this information will capture in a data set level? The 101 subject is exposing to the drug A, drug B, and placebo. 102 subject is taking placebo, drug A, and drug B. Okay, here one subject is taking multiple drugs. One subject is taking multiple. So by seeing your data set also, we get to know whether it's a parallel study design or crossover study design. Okay, so in a parallel study design, one subject have only one drug information. In a crossover study design, one subject have a multiple drugs information. Okay, like this, we have a 2 types of study designs will be there. Parallel study design, crossover study design. Okay, clear? Here, any doubts? Nothing? No. Understand the difference, right? So the next definition, site. What is site? Actually, I told you, right? What is site? That is not hospital only, right?
Where trial has to conduct, sir.
Hospital, but where the clinical trials are happening. Not all the hospitals where these clinical trials will happen. So those we call as a site. The hospital where these clinical trials are happening, we call the site. Okay. Next. Multi-center study. What is multi-center study? It's if these clinical trials will be conducted in a different cities or places in the same country. Let us assume if you choose in India, they are conducting the clinical trials in Hyderabad, in Chennai, in Bombay, in Delhi, in different places, they are conducting the clinical trials. If it is conducting like this, it's a multi-center study. Similarly, multinational study. Multinational study. Now this one. So same, the clinical trials will be conducted in a different countries. Okay, so they're conducting the clinical trials in India, in Japan, in USA, in Australia, different countries, they're conducting the clinical trials. Okay. Next. Next is blinding. Blinding or masking. Blinding or masking. Okay. Blinding or masking is called nothing but blinding is nothing but hiding the treatment. Hiding, that means what treatment we are giving, the sponsor is giving, will hide to the different group of people. So based on this hiding, this blinding was divided into 3 categories. There are 3 types of blindings here. Okay, one is single blinding. Second is double blinding. Third is triple blinding. Single blinding, double blinding, and triple blinding. What is single blinding? Okay, I was telling that 3 different groups, right? There are 3 different groups will be there. One is patients, that's called subjects, and investigators, that is principal investigators, Principal investigator is nothing but doctors, principal investigator is doctors, senior doctors, or biostatisticians. So three different groups will be there. Now, if you are hiding the treatment to the only patients, means only patient doesn't know. No to the patients, yes to the doctors and investigators, principal investigators. So if you are hiding the treatment only for patients, then it's going to be a single blind. Means patient doesn't know which drug is taking. Which treatment is taking. In a double blinding, patients, investigators, and principal investigators. So your investigator doesn't know. No to the patients, no to the investigators. Only principal investigator have information which drug is taking. So we're no to the two groups. So it's a double blinding. In a triple blinding, okay, we are no to all the three. Patients, investigators, and your principal investigator also present. Now tell me then, in a triple blinding, who have a record? Which treatment is taking? Subject. Who going to have information?
Sponsor, sir.
Correct. The sponsor have a record, right? You're going to have a record in a triple blind. Important. Okay. Next. The last one is open label or unblinding. So unblinding means, yes, everyone knows. Even patients, investigators, and principal investigators. Yes, to everyone. Unblinding. Clear? Like this, there are majorly three types of blindings. We have single blinding, blinding,
Sir.
and triple blinding.
So could you please explain single, double, triple blinding? I just missed it.
History of drug.
See, blinding is nothing but hiding of treatment. One means not disclosing or not informing what treatment we are giving to the patients. Okay. So if your study is a single blinding study means, so in that study, the patients doesn't have a information. We are no to the patients. It means you will not inform what drug we are giving to the patients. So patient doesn't have a information, but investigators and principal investigators have information. It's a single blinding. If you are not hiding, if you are hiding to the patients and investigators, both means it's a double blinding study. Means only principal investigator will have the information. In a triple blinding, all the three people, patients, investigators, and principal investigators, all the three people doesn't know. You're not informing to all the three different groups. We're hiding the treatment for all the three categories. Then it's going to be triple blind. Finally, unblinding is nothing but everyone will know. Patients, investigators, and principal investigators. So it depends on your study. Your study is a single or a double or triple or unblinding study. Every study has a different thing. Clear? Any questions? Anyone?
Sudarshan, what would be the reason for conducting a blinding study?
So this is their sponsor decision, right? So they want their intention is to expose their study drug. They want to inform, they can inform. If they don't want, they don't. So the intention is nothing about it. It's about the confidentiality, about the risk how the drugs they expose. Okay, some people are not ready to take the drug also. Okay, it's about the sponsor decisions. Okay, their internal things.
Wouldn't it be dangerous on the subject for blinding study if they don't know what the drug is?
So that's about while he has to sign the informed consent form, he has to think and he has to sign. Okay.
Okay.
So we cannot take that decisions, right? It depends.
Okay.
If that dangerous, why it will come in the clinical trial stage? It will stop in the pre-clinical trial stage itself. Okay.
Okay.
And then everything will this information cannot be hidden by anyone. Okay, it should be informed to the FDA also. Okay, FDA will aware everything. The sponsor is doing which study and what they are following.
Okay.
Okay.
Sir, I have a doubt.
Yeah.
Sir, these is they have to register about the clinical trial, sir.
They has to register about the clinical trials. Obviously.
Okay, okay. Through whom, sir?
So they expect to authorities. Okay. You are doing the clinical trials in India means for India. In India, they has to whom you have the higher authorities, higher affiliates will be there, right?
Okay.
For them, they has to inform. Without their knowledge, you cannot do the clinical trials in any
Okay.
country or any city.
Okay, okay.
Okay.
Hmm.
Respective authorities will be there. For authorities, they has to inform. Then they has to do.
Okay, sir. Sir, is there any sites, sir, that we can see any the sites, sir, regarding this one?
No, regarding this one, we don't have that, but we have information only for the CDISC only we have.
Okay, okay.
CDISC website will be, if you just Google it, you will get it. The CDISC organization is. For the CDISC, you have rules and set of rules and regulations.
Okay, sir.
Having the information there.
Okay, sir.
Few more definitions also there. We'll discuss those definitions in the next class. Okay. Chalo. So that's it I have for today. Any questions, anyone?
Hi, Sudarshan. It's just for the information only. So after we got the all the guidelines since 1996, you said?
1997.
1997. So how much time now it taking for the medicine? Previously, it was 2 to 8 years.
No, now also it is now that is today it is now only.
Oh, okay.
More than that. Okay,
Makes sense.
fastest vaccine we received for this one only, COVID only. Any other, if we take any vaccine or any medicine, minimum 2 to 8 years. Okay.
Okay.
So for our knowledge is why, as I mentioned, this point is why as a programmer we need to know means you are working for one project means the project will not over in 1 year, 2 years. Okay. You are joined for one company. If you're there in that company for 5 years, also the project will be there for 5 years.
Yeah, and that's the same reason I asked the question as well. That like
So you won't jump to any project. You have to work on the same study, same work, daily, same thing. Only data will refresh. We'll do the HDTM, Adams and tables. HDTM, Adam tables. HDTM, Adam tables. Repeat. Same study, same data, same thing. They'll refresh the data. You'll get to know. Okay, going forward, I'll explain the flow and all. Once you understand the flow, workflow, then you'll understand how is your work culture also. Okay.
That means every day we have to write only these things only, sir.
Same things.
Just minor modifications only.
Only one time you'll write a program. Next time onwards you'll rerun the programs.
Okay, okay. Just minor.
Understand, okay, going for in the next class you'll understand. Next class I'll explain the workflow.
Okay.
The workflow you'll understand, okay. What's your work will be, where it started, where it is ended. In the flow, what is the thing I'll explain. You will have a knowledge. Now we are
Okay,
just starting only, right? Just to have a knowledge.
Okay, sir. No problem.
Okay. Okay, then fine, then anything.
Recording stopped.
Okay, fine then. That's it. We'll continue the next class.